afternoon, everyone. I welcome you all on the behalf of Biofootprints in this wonderful talk on the topic From DNA Structures to Biotechnology, Breakthroughs that Shaped Life Science Research by Dr. Timir Tripathi. Dr. Timir Tripathi is a senior assistant professor and principal investigator at the Department of Biochemistry, Northeastern Hill University, Shillong, India. He holds a PhD from the Central Drug Research Institute, Lucknow, India. He has performed pioneer work on protein-substrate interaction and dynamics and understanding the roles of non-catalytic domains in regulating the catalytic activity of proteins. His primary focus is on the structural and functional characterization of key drug target proteins from the helminth parasite, Fasciola gigantica, for developing therapeutic strategies. Dr. Timir has received several awards, including Professor B.K. Bachawat Memorial Young Scientist Lecture Award 2020 by the National Academy of Sciences, India. He has also received ICGEB Young Scientist Award 2019, ICMR Shakuntala Amirchand Prize 2018, BRSI Malviya Memorial Award 2017, DST Fast Track Young Scientist Award 2012, DBT Overseas Associateship Award 2012 and Dr. D.M. Bose Award 2008, etc. He is also an elected member of National Academy of Sciences, India and the Royal Society of Biology, UK. We welcome you, sir. Thank you for taking out time to give us the lecture. You can start now.
Okay. So first of all, I would like to thank Biofootprints and the organizers, Professor Sujata ma'am, Pradeep and Akshita, for inviting me to deliver this talk. Without wasting any time, I'll just share my screen and start presentation. You have to enable myself. Akshita?
Sir, you are already...
No, it is disabled participant screen sharing.
It will be done from Madam's account.
Now it's there. Now it's there. I think you can see.
Yeah, yes, sir. Yes, sir.
Can you see that? Okay.
Yes, sir. Yes, sir. Your screen is visible.
Very good. So I will do well. I have some. There are internet issues in Northeast, so I will keep my screen off. Okay. Now, can you see the slides?
Yes, sir. Yes, sir. You can turn the presentation mode on.
Is it okay?
Yeah, we can see. Yes, sir. Yes. Sir.
Okay. So just relax. Okay. So today we will be discussing about a story from the discovery of the structure of DNA to biotechnology. So we will cover about 70 years of research in the next 45 minutes or so. I will try to be as simple as possible. And in these 70 years, there is not only research in biotechnology. There are research in several fields, including structural biology, drug discovery, and so on. But today's discussion, we will be focusing on the research and development that have shaped life science research in the field of biotechnology and genetic engineering. So let us start. Okay, everything started with the discovery of the double helical structure of DNA in 1953. So the twisted ladder-like structure of DNA that we all are familiar, we call as double helix, was determined by Watson and Crick in 1953, and this was the starting point of modern biology and genetics. In fact, this was one of the most important early milestones that described genetics and was the backbone of many future discoveries that we will be discussing in the next some slides. So this discovery is considered one of the most significant early events in the field of genomics and genetic engineering. Yet a person is often missing in this narrative, Rosalind Franklin. Watson and Crick, they owed their discovery to their colleague Rosalind Franklin, without whose X-ray diffraction image of DNA in early 1950s, they would have never considered the double helical structure of the DNA structure. And for the structure of DNA, Watson, Crick, and Wilkins, they got the Nobel Prize of 1962. So we need to remember briefly Rosalind Franklin. Maurice Wilkins was a scientist along with Rosalind Franklin. They worked together on different aspects of DNA structure. And Rosalind Franklin, after several complicated analysis with X-ray crystallography, she discovered, and she was the first to state, that the sugar phosphate backbone of DNA lie on the outer side of the molecule. And she also determined the helical structure of DNA. Watson and Crick were awarded Nobel Prize in 62 along with Wilkins. Disappointingly, Rosalind Franklin, who actually contributed to the double helical structure of DNA, she died before that age. She died at the age of 37. And at that time, Nobel Prize Committee did not permit a prize to be awarded posthumously. So she did not receive a Nobel Prize. But this discovery of DNA resulted in an explosion of research into molecular biology, genetics, and genetic engineering, providing the foundations of biotechnology development. There are several stories around Rosalind Franklin, which we should discuss, but this will not be a time. It needs a separate an hour lecture. This was the DNA structure, the X-ray diffraction pattern that Rosalind Franklin determined, and this is the notes of by her hand. And in fact, she was having several issues with her senior colleagues. One day she wrote a letter. This is a letter that she wrote with her hand. She was so depressed that she wrote, "It is with great regret that we have to announce the death on Friday 1952 of DNA helix." So these are another stories which we can discuss sometime else. So for Crick, when he started working on central dogma of molecular biology, there are 4 kinds of information that existed. DNA can form DNA through replication. DNA can form RNA, first step of protein synthesis. RNA can form protein, second step of protein synthesis. RNA can form RNA. This is also possible in case of RNA virus. Can you just mute your audio? It's very difficult. Okay, so these information was available with Crick at that time, but there are 2 steps which was not evident, but Crick thought they were possible. So these dotted step, the Crick thought that it is possible. That is from DNA to protein and from RNA to DNA. So this also should be dotted. This and this were possible, but there was at that time there was no biological evidence that this is possible. So Crick, he admitted that there was no evidence at that point, and it was very speculative. But he defended his approach by pointing that, like a cosmologist had no qualms about building theories without experimental data. So one has to hypothesize. So Crick concluded a genetic material must carry out 2 jobs. First is duplicate itself, and the second is control the development of rest of the cell in a specific way. In 1958, Arthur Kornberg, who has been working on project of DNA synthesis from early 1950s, by 1953, what happened? He could synthesize all the 5 nucleotides in his lab, AGCTU, and then he turned his focus towards the remaining factor required for DNA synthesis. So that is the enzymes and all. So he isolated DNA polymerases from bacterial extract and synthesized DNA in vitro for the first time. Now, interestingly, interesting story with Kornberg is his initial paper, where he reported the DNA synthesis data. He submitted that to JBC, and some reviewers refused to term the product as DNA. They suggested to use the product polydeoxyribonucleotide. Even one reviewer asked for the genetic activity, proof of genetic activity of that molecule. So what Kornberg did, he withdrew all his papers until a new editor of JBC was appointed in 1958, and then he published those work in JBC. So Jacob and Monod then in 1959 figured out how bacteria controlled the production of an enzyme, beta-galactosidase. He identified the system of feedback, negative regulation that we know as lac operon. And this was the first model for the control of protein production. So what their work concluded was they discovered regulatory genes, operator genes, regulation of the structural genes, and all these, and they revealed that genome contained not only series of these blueprint gene sequences, but also a coordinated program of protein synthesis to control its execution. So then started the era of 1960s with the origin of modern genetics and genetic engineering. Now it brings us back to Silicon Valley. So 60s saw an explosion of research into the structure and function of prokaryotic and viral genetic material. In 61, Marshall Nierenberg, Khurana and colleagues, they deciphered the genetic code. All of you know, they determined the genetic code and showed how the nucleic acid bases, that is made up of A, G, C, T, they determine the sequence of the 20 amino acid in different combinations. So each codon, it is specify a specific amino acid which is added during protein synthesis. So they reported the genetic sequence of 3 nucleotides, each say phenylalanine UUU. Like that, they reported the genetic sequence of 3 nucleotides, that is they called as codons, and that decide all 20 amino acids. So Hawley, Khurana and Nierenberg, they shared the Nobel Prize in 1968, where Nierenberg and Khurana, they got this prize for genetic code, while Hawley, he prepared the first tRNA molecule and he got for that. Okay, so I do not know how to remove this. Okay, anyway, in 1962, jellyfish system was a protein was identified to study the location of any protein inside the cell. So there are organisms which produce fluorescent protein, like green fluorescent protein, which emits shimmering light. So this formation of this gene, green fluorescent protein, is regulated by a particular gene. That gene you can take from jellyfish and incorporate in other organisms. So since GFP can be linked to other proteins using genetic engineering, it became very important tool to understand the biological process of cells. So it was Shimomura who first isolated this GFP from jellyfish in Aquaria and Victoria, and found out that this protein glowed green when illuminated with ultraviolet light. So this accomplishment was very important in the field of genetics, because once you fuse this GFP to another gene, that protein produces a protein of interest, you can determine which cell expresses the target gene and what is the exact subcellular localization of that protein. For that, he also received Nobel Prize in 2008. So in the era of 1970s, genetic engineering boomed, and this decade demonstrated the fundamental achievement that shaped the genetics and genetic engineering for all future scientists. So in 1970s, the discovery of restriction enzyme and DNA ligases, you know, restriction enzymes, we also called as restriction endonuclease. It is an enzyme which is produced by bacteria and that cleaves DNA at specific sites. So today, the scientists depends on this restriction enzyme for virtually any process that involves DNA manipulation, say cloning, to understand a diagnosis of hereditary diseases, paternity testing, genomics, creating genetically modified organisms, biotechnology, etc. In fact, without the discovery of restriction enzymes, the field of recombinant DNA technology would not exist. So after their discovery, Smith, who was from Johns Hopkins, he purified the first site-specific restriction enzyme from the bacterium Haemophilus influenzae, and he identified the exact six base pair location where that Hindi to that enzyme that cleaves. So Werner Arber was there. He noticed that there are certain bacterial strains which can fight the bacteriophage infection by chopping off its own DNA. So he thought that why these not restriction enzyme, they cleave off their own DNA. So he hypothesized that these bacteria, they produce two types of enzymes. One is a restriction enzyme and one is a modification enzyme. So restriction enzyme, it cuts the foreign DNA, while the modification enzyme recognizes the host DNA and protect it from cleavage. So what he did, he proved this hypothesis. He took unmethylated two types of DNA, and he proved his hypothesis where he took these two enzymes, restriction enzyme and modification enzyme, and when he used it to cut it, then the modification enzyme, which was methylase, it protected the DNA of the bacteria, while the restriction enzyme, it cleaved off the DNA. So he identified how this happens. And the understanding of how this restriction enzyme cut the DNA and how the host DNA works provide the basis of modern genetic engineering. So later Arber, Nathan and Smith, they were awarded Nobel Prize in 1978. So in 1972, Paul Berg, he already had this restriction enzyme. So what he did, he took restriction enzyme to cut the DNA into smaller fragments, and then he used an enzyme called as ligase to make a hybrid molecule. And this was the first recombinant DNA molecule that Paul Berg made. So this recombinant DNA molecule has been created through the combination of elements from different organisms. So this is from some organism and this is from another. So by genetic engineering, by introducing genetic engineering from one organism to another, this discovery showed the first principle of modern genetics. In fact, in the same year, Cohen and Boyer, they were two scientists, they showed that the key to this recombinant DNA is that it can replicate itself. And despite you introduce it artificially inside a cell, they can replicate themselves. So the development of this recombinant DNA was considered as a fundamental research in the field of genetic engineering and was the biggest stepping stone towards the creation of recombinant DNA. So Paul Berg later on 1980, Paul Berg along with Max Gilbert and Singer, he received the Nobel Prize. This Singer is the insulin. So by all this time, when genetic engineering experiments were happening, and all these experiments were great for science, But with the achievement of the scientist or by manipulating DNA, it was evident to the National Academy of Sciences USA that there may be ethical dangers which is associated with this experiment and which will develop in future. So they proposed a temporary moratorium on this genetic experiments in 1974. However, what happened? Paul, he realized the importance of this field. He organized one conference, ICMR conference in 1975, which was attended by over 100 scientists in that field. And at that conference, many of the ethical ideas surrounding the genetic engineering experiment were developed and they were agreed upon. And in fact, still adhere to date with modern genetic engineering. So there was also a scientist in that meeting, Joshua Liederberg, who has already got 1958 Nobel Prize for bacterial conjugation. He was a Stanford professor and he emerged as a strong voice highlighting the potential of this technology in curing diseases. So his optimism and say foresight, they made people control their fear about this technology. And then Liederberg, he argued that these recombinant DNA technologies will have a very successful future, and it will help in the growth of science and biotechnology. Next year, 1975, hybridoma technology, it revolutionized modern genetics. As you all know, there is human B cells has an amazing ability to clone itself. It can clone itself and produce exactly identical copies, antibodies known as monoclonal antibodies, and these monoclonal antibodies help in fight immune-related diseases. So Kohler and Caesar, Milstein, these two, they found a way to mimic this effect by using myeloma cells. So this is a type of cancer. Myeloma cell is a type of cancer of plasma cells. They mixed it with B cells to produce the antibodies using certain method. And this system was known as hybridoma technology because it involved cells hybrid, two cells, different cells hybrid, to produce set of exactly same monoclonal antibody. And this technique to produce monoclonal antibodies laid the foundation to explore antibodies for diagnostic purposes, therapeutic purposes, and other scientific application, immunology-based treatments, and so on. Maxam and Gilbert in 1977, they were from Harvard, and Frederick Sanger, he was in UK MRC, Medical Research Council. They developed research for sequencing DNA at that time. And Frederick Sanger, he used this technique to determine the DNA sequence of a first bacteriophage. This was 5X174 bacteriophage. He determined the sequence of this organism. And for that, they got the Nobel Prize of 1980s. So now you can see in each decade, huge amount of scientific developments are happening. In 1980s, the 1980s turned many genetic engineering experiment into real world product and ideas that changed the landscape, which will be coming in the next decade, 1990s and 2000. So in 1980s, the scientists focused on vaccines, they focused on treatments, and this era was displayed as the scientists' dedication to solve human problems using humans' own DNA. 1981, the first transgenic animal was created. Transgenic animal means any animal that has a gene from any foreign organism, which was deliberately inserted into its genome. So today, transgenic animals are very common. people create transgenic animal using modern genome editing method. But the first transgenic animal was developed by Wagner. This is Jolick and Wagner at Ohio in 1981. And Wagner, he transferred a particular gene of rabbit. He took this particular gene of rabbit into a mouse and to create a world first this transgenic element. He took this DNA and then used micro injection to inject inside the mouse and create a transgenic element. So this discovery and the Wagner's use of process to create this transgenic animals revolutionized biomedical research, and they provided a method to create future transgenic animal experiments. So in 1982, Dennis Cleve, he was a scientist from Genentech, a biotech company. He recombinantly produced the first genetically engineered drug, and that was insulin. So for the better part of the century, insulin was used to treat patients suffering from type 1 diabetes. However, insulin was harvested from animal. And this animal insulin, they were quite similar to that of human, but there were slight differences. And in 1970s, this insulin was, if you want to create a single pound of insulin, it required around 8,000 pounds of pancreas, and which comes from around 25,000 animals. And this required to meet the demand of US only, it required more than 50 million, sacrifice of more than 50 million animal annually. So a synthetic alternative was necessary. And when he created this genetically engineered insulin, it was considered as a defining moment for the concept of the application of genetic engineering genetic engineering drugs for human consumption. So US FDA in 1982, they approved this insulin and for the treatment of diabetes. Okay, so next year, the discovery of PCR, the polymerase chain reaction. Just one second. Yes. So the discovery of PCR was integral in later DNA experiments. PCR is used to make copies of specific DNA segment. So by implementing chain reaction, any DNA sequence can be amplified from a small number to a very large, I mean, millions of copies of number. So this discovery of the PCR technique was done by Carey Mullis, and it made DNA very accessible to scientists by reducing the time to clone DNA. So there were restriction enzyme, there were ligases, but to clone you need large amount of DNA, which it was difficult to obtain. And Mullis's experiment using this PCR made possible to make it very quickly and cheaply. So this technique made DNA experiment more efficient and more doable. So by now, next year, the first recombinant human vaccine for hepatitis B, people were working on that, and then this was developed. In fact, the first vaccine for hepatitis B from animal, it was developed in 1963. But the first recombinant vaccine was produced by Valenzuela. He used yeast Saccharomyces cerevisiae to create this hepatitis B vaccine, and this vaccine immediately became market standard, and the blood-based vaccine, the animal-based vaccine was removed from circulation. So, in fact, by the method what Valenzuela used to create vaccine, many vaccines that we still use such as papilloma virus or whooping cough or pneumococcal or meningococcal viruses, they were created using the same technique what Valenzuela developed. So by this time now, in 1988, it was the first time when a genetically modified crop, it actually appeared in the fields of United States, and that crop was BT corn. BT corn, it is named BT corn because it contains gene from bacterium Bacillus thuringiensis, and this corn was able to increase the yield by discouraging the pest. The pest, normal crop, the pest can infect, but this BT crop corn, the pest were not able to infect. Similarly, BT cotton, this is normal cotton, this is BT cotton. So BT cotton is a insect resistant transgenic crop which was designed to combat the bollworm. So how this BT cotton was created, the scientists took a particular gene which coded for a protein from the bacteria Bacillus thuringiensis and introduced it in the cotton. So now once this transgene was inserted in plant's genome, this plant produces toxin, which actually the plant will not produce, but since we have introduced from Bacillus, it will produce. And once this the insect will eat this, they will die because it digests the intestinal lining of the insect. So China first planted BT cotton in 1997 in response to a cotton outbreak, which the farmers were not able to control using pesticide. And in 2004, the United Nations formally endorsed these BT crops or biotech crops as a way of supporting the farmers of a developing nation and to solve world hunger crisis. So 1990s, say from 1990 to 2003, the Human Genome Project succeeded in mapping the human genome to around 20,000 genes. Their genomic loci was documented, and these 1990s was a period and that proved that the work what people were doing with genetic engineering and genetically modified organisms, they were not only valid, but they were very necessary. So the Human Genome Project, it was launched in 1990, and the goal was to sequence all the 3 million base pairs, 3.4 million base pairs, sorry, 3.4 billion base pairs that are found in the genome of humans. And this knowledge is very important for research into genetic disorders and possible genetic solutions for these disorders. So this project was led by James Watson, and the initial target was 2005, but it was completed in 2003 only. So during this 1990s to 2003, there was a large amount of organisms that were sequenced. The first organism which was sequenced and the first free living organism was Haemophilus influenzae. Then the first eukaryote was in 1996, Saccharomyces. Genome of nematode 1, C. elegans in 98, fruit fly Drosophila melanogaster 2000, mouse 2002, and rat 2004, and chimpanzee 2005. So you can see from started in this 10 years from the first genome to we came down to chimpanzee and human, very complicated and large genome. So presently, thousands of other organism genomes have already been sequenced, they have been annotated, and the technology is such that human genomes can be sequenced in a couple hundred of dollars. So extensive bioinformatic resources were also developed that allow the genome sequences to be stored and explored online. As you know, as of now, more than in fact 3 million SARS-CoV-2 genomes are available in the databases. GISAID database is there, which CoVGlu database. which has more than 30 lakh human genomes. See, SARS-CoV-2 genomes available. So coming down now to CRISPR. So many people, they regard CRISPR to be pioneered by Doudna and Charpentier. So they got the Nobel Prize also. And they believe, people believe that Doudna and Charpentier, they pioneered the CRISPR. However, the principle of CRISPR was actually discovered by Mojica while he was working with a bacteria in the marshes of Santa Paula, US, where he noticed that the DNA of the bacteria repeated several times with regular spaces in between. So this was in 1993. So from 1993 to 2003, Mojica continuously was looking into these sequences, repeated sequences, and how they were present in that DNA and how they were matched, and he matched these DNA sequences with the viruses which were attacking this bacteria. So now this principle led to many advancements in DNA study over the next 10 years, and those techniques which brought CRISPR into existence, CRISPR gene editing. Okay, this was the biggest story, Dolly. This was one of the biggest stories spread through the public that science has ever seen. Dolly, the sheep was cloned in 1996. The cell was taken from the mammary gland of a 6-year-old Finn Dorset sheep, and the egg was taken from a black Scottish sheep. So she was born to her surrogate mother. This is her mother, and this is Dolly. So she was born to her Scottish mother. Scottish was black on 1996, and Dolly's face was white. And this was the first time that she was a clone, because if she was genetically related to her surrogate mother, she would have had black face. Later on, Dolly was diagnosed with tumor and was put to sleep in 2003 at the age of 7. And his body, and Dolly's body was kept in the National Museum of Scotland by the Roslin Institute, and where it became one of the most popular exhibits. So I'll not go into the detail of this. I'll just come down to this. In 2003, the first cloning of an endangered animal took place. So this was banteng. Banteng is a wild bovine which is present in Java. So this is an endangered animal, and our scientists were able to clone this banteng, and this was in fact a great example of how cloning can help save species that are endangered or that are going to exist in the near future. So in 2000, the progress in mapping of human genome at the end of 2000 was a great way to finish off the whole millennium, and it showed incredible promise for the years to come. So during this period, new methods were developed, new discoveries were happened, which showed that which opened the door for new high quality experiments. In 2001, the first gene therapy. What is gene therapy? Gene therapy means it uses a section of DNA, a gene, which is used to treat or prevent diseases. So gene therapy approach is used to treat, cure, or even prevent diseases by changing the expression of a person's gene. So currently this field is growing very fast. There are clinical trials for many types of cancer and other diseases. So first gene therapy that was approved by the US FDA and was sold as an anti-cancer drug to treat chronic leukemia. And this drug, Gleevec, this was the drug. It is still used to treat cancer patients today. So this drug they target some platelet-derived growth factor. Again, I will not go into the detail of this, but this was the first gene therapy which was approved. Now, taking a more fun approach towards science, towards genome editing, there were scientists called Bill Blake and Crockett. They developed the first element for sale as a pet that has been genetically altered. So they named it as glowfish. So this glowfish was the first pet which was genetically altered. So glowfish literally means the fish that glows. So this fish uses a naturally occurring fluorescence phenomena. The gene was inserted so that the fish was able to glow. So this was more of a fad than a lasting scientific achievement. This marked the start of an era where public was more open to the idea of gene-edited organism. Okay, in the year 2003 only, remember, it was the Human Genome Project was completed and confirmed that humans have around 20,000 genes, and the genome was sequenced with 99.99% accuracy. Okay, so in 2006, there was a breakthrough using gene editing to find cancer treatment. There was a drug, Gardasil, which protect infection against two types of HPV, human papilloma virus, that caused majority of cervical cancer. And this was the first preventive cancer vaccine to ever reach market. And the popularity of this vaccine was substantial, though considering the fact that it was a recombinant vaccine, but still the popularity was very high. And till date, this HPV vaccine is the only vaccine, preventive cancer vaccine. By this time only, another popular discovery was in the field of stem cells. iPSC, induced pluripotent stem cells. They are type of pluripotent stem cells that can be directly generated from a somatic cell. So this research was done by Yamanaka from UCSF. He introduced the first technology. I'll not go into the detail, but this technology revolutionized scientific research in the coming years. So 2010s, the new era of FDA-approved therapies. In this decade, development therapies that has been studied from last 40 years were finally being approved for human, such as a discovery of CRISPR and all. In 2010, the world first synthetic life form was discovered. So the scientists created the first life form, synthetic, which technically means a living organism was created instead of evolved or born. So they used an organism, Mycoplasma mycoides genome, and this was done by Craig Winter, and they transplanted in M. capriculum recipient cells to create a new M. mycoides cells, and that these cells were completely controlled by the genes of this and not this, completely controlled. So the DNA code of mycoides, how it was written? It was written on a computer. Just like you design a primer, just like that, code was written and then created artificially and then inserted in a bacteria. So this marks an important stage in the field of biology, as it proved that the life can be created synthetically, and this has great application. So welcome to the world of synthetic bio, or people called as synbio, where possibilities are limited only by imagination. So now you can imagine a future where say, synthetic jellyfish. They roam around the waterways looking for a toxin to destroy those toxins. And say eco-friendly plastic, fuels, which are harvested from yeast. And even say bacteria, viruses, you can engineer them they can program them to kill the cancer cells. Or even like electronic gadgets, which can repair themselves as like a living organism. So this can happen. 2012, Doudna and Charpentier elucidated the mechanism of CRISPR. Not without going into detail, just understand that CRISPR can precisely cut DNA and manipulate the DNA as you require, and this showed endless potential in the field of medicine, agriculture, biomaterial. Today, CRISPR is used for developing cancer treatment, obesity, tackling obesity, and much more. So whether it is diagnostic treatment or something else, there are new discovery coming in CRISPR every day, even today. So in 2013, Feng Zhang from MIT, he demonstrated the use of CRISPR outside the world of bacteria. So initially in 1993, as well as in 2012, they demonstrated the application of CRISPR in bacteria. Now, Feng Zhang, he showed the importance, the role of CRISPR in genetic manipulation of eukaryotic cells. So he developed tools that are broadly applicable to study genetic diseases, diagnostic, therapeutics, and even nervous system function, genetic disorder, and so on. So in fact, with these techniques, the journey of genome engineering has just started. So there are discoveries which are happening throughout the world in the field of agriculture, energy, environmental science, animal husbandry. There is a lot of opportunities. And with the development of sophisticated high throughput method, the world is continuously, the scientists are continuously trying to make the world better by driving the genome engineering technology. Now quickly, I believe I have crossed almost 45 minutes I'm reaching. I'll try to finish that in next 6 to 7 minutes, if I'm allowed.
Yes, sir, no problem. Please continue.
Thank you. So in 2014, there was a scientist again from MIT, Esveld. Now he took the idea of a CRISPR to a whole new level. When he realized that the ability of CRISPR to create gene drives. Now, what are gene drives? Gene drives are genetic elements that cheat evolution by biasing their transmission to offspring beyond Mendelian laws. So this gene drive is a genetic engineering technology that propagates a particular set of genes through a population by altering the possibility or probability of a specific allele will be transferred to the offspring from the normal 50%. So generally it is 50%, no? It is 50% possibility, it is 50%. So here it will not be there. But gene drive, here it will be 50%. This side, it should be only this one. So you are changing the genes, the altered gene is always inherited. So now these genes, once you change these genes, and the altered gene can be introduced in population. So gene drive may be used to spread a particular genome changes that target the wide population over many generations. Now try to understand the potential of this in eradication of malaria and other vector-borne diseases, and as well as saving endangered species. However, it is important to understand that these have ethical implications. Even introducing in ecosystems may be catastrophic. So still he hasn't released these mosquito. But last year he gave an interview where he told that he can prepare mosquitoes that will be suitable for field test in less than 1 year. But he is not in rush to release them and not trying to do anything foolish outside the ethical consideration. So in 2015, the first genetically modified salmon, this is salmon, general salmon, and this is Aqua Advantage salmon, the big one. Aqua Advantage is the name given by the company. So the company was Aqua Bounty. So they were first to sell this genetically modified salmon for human consumption. So this Aqua Advantage salmon is genetically engineered, where a typical growth hormone was replaced, but from some other salmon. So with a specific promoter. So this gene, it enabled the genetically modified salmon to grow bigger in size as well as year round, instead of only spring and summer. So these salmons are available only in spring and summer. This is available year round. So this was sold in Canada. In 2017, the first, in fact, two CAR T-cell therapy was approved. One was for acute lymphoblastic leukemia for children, and other was for advanced lymphoma for adults. Okay, so in simple words, what happens in CAR T-cells is you isolate the T cells from a patient, take that T cells, modify it in vitro in a test tube using a antigen receptor, CAR, chimeric antigen receptor, and then they are amplified and then they are put back again to the human. And now these modified CAR T cells, once they are inside, they play specific anti-cancer effect. So now this was a huge step for CAR T therapy, which if treatments prove as effective as it is expected, it will likely replace chemotherapy in the future as the primary form of treatment. In fact, these CAR T cells are not toxic to body because they are taken from person's old T cells, and they have in fact demonstrated this. This is an example what happened. Complete recovery by a small child. Complete recovery within 10 days. It showed complete abolition of cancerous tumor from the body. Okay. So in 2017, scientists encoded a movie inside a E. coli genome. Let me try to play it first. So this is a small. Yes, this is a small movie. The scientists, what they did, scientists at Harvard University and other institute, they built the first so-called molecular recorder using this CRISPR gene editing. So now the aim of this molecular recorder is to record the data in the genome of living cells. So once you record this data in the genome of the living cell, you can access it later. And this will provide insight that could help disease or even deepen our understanding into biology. So to prove this, this is a very far out possibility. What the scientists did, they successfully encoded complex information that included the image of one hand, a sequence of a galloping horse, and this was the first motion, and just they incorporated that into the DNA of bacterial cells. So this work demonstrated that this system can capture and stably store practical amount of real data within the genome of population of living cells. So see how science is progressing. 15 to 19, human genome editing. There are many things happened in this 4 years. Chinese researcher Huang, he edited human embryo, which was originally rejected by scientists because it did not follow the ethical rules of science, but later it made into, somehow he made into publications in other way. what Huang did, he altered the germ-line cells which affect the heredity. So his experiment aimed in fixing a error which was there in a gene in certain blood diseases. But however, since he was manipulating in humans, human embryo, it was not considered ethical and became a huge controversy. He was in fact he was jailed, and then later he was recruiting. This is the whole story, how he was what he was doing. He planned, then he recruited couples, twin girls were born. MIT Technology Review Journal, it produced magazine, it revealed the existence of this research program. Press went, and then he offers this detail in a conference in Hong Kong and was criticized. China ordered investigation, and while this CRISPR based editing was approved, but he was censored by Chinese Health Ministry, and WHO during this period they set guidelines for human gene editing. And during this August 2019, third gene edited baby was born, and CRISPR edited human trial started. But in 2019, he was sentenced for prison by Chinese court, which is still in. So several things happen. Whether it is ethical or not ethical, we should not right now. We should not delve into that. In 2020, now we are coming just 2 more years. 2020, CRISPR gene editing for sickle cell anemia. All of you know, sickle cell anemia is a disease where genetic disease where there is a defective hemoglobin formation. So the RBC becomes deformed sickle shaped, and due to that, they get jam inside the blood vessel. They cause pain, organ damage, and also lead to death. So this was a scientist, Heider Frangol, in a conference at 2020. He presented his data showing that this lady who is going treatment, and there were other two also treatment of two patients for treatment of beta thalassemia, and he showed that they are recovering well. So he used CRISPR gene editing for treatment of these people. As you know, in COVID-19 pandemic, also one of the method researcher they are using to is the CRISPR gene-based editing for diagnostic gene as well as detection, diagnostic, as well as for treatment. So gene editing could be potentially used, theoretically used on the genome of the SARS-CoV-2 to make it harmless. So people have used that to develop better testing kit, or even it could be theoretically, it can be used to edit human genome to prevent people from being infected by the viruses. And in fact, this was a paper which came in iScience yesterday, showing the diagnostic potential of CRISPR system for detection of several viruses. In 2020 only, there is an interesting story. So what happened in an Indonesian city, Yogyakarta? So there was a very high increase in dengue fever. So what happens? Researcher, they released one mosquito which were infected with Wolbachia. So Wolbachia carrying mosquito were released for around 6 months to start to study this the disease severity or disease spread between people. So where it was spread, it was observed, and these were all genetically modified. So before this, rates of dengue in this period were very less, but in compared to those where they did not receive the mosquito. In 21, the first genetically modified mosquito, they are released in the US in open air, and this aimed to suppress population of wild Aedes aegypti mosquitoes. This carries several virus diseases like Zika, dengue, chikungunya, yellow fever, etc. So in fact, this year only, 2022, researchers are working on the same product. They have told that they will release bioengineered male this Aedes mosquito, which don't bite. Male do not bite. So to mate with the female wild population, and the female is responsible for biting and transmitting disease. So now these genetically engineered males, they carry a gene that passes to their offspring and kill female progeny in their larval stages. So the male offspring will not die, but instead will become carrier of the gene, and it will pass on to future generation. So as more female will die, the Aedes population should dwindle. So these genetically engineered mosquitoes are alternative to insecticides, which are used heavily in US and to control insect population. Last year only, there was first evidence that in vivo gene editing in a clinical setting is also feasible. And this happened with no early sign of severe adverse effect. This is an ongoing trial which is going on to treat transerythrin amyloidosis, amyloid-based diseases, a monogenetic disease. Last week only, the researchers in the China government has approved gene-edited crop. So in 11th February, the researchers in China, in fact, they have welcomed the government approval of gene-edited crops. So however, China's rules are much more conservative than that of the US. So which do not regulate plant that incorporate small changes, and that happen naturally. But they are more lenient than the European Union stance. Okay, there are several things which are happening and continuously happening. I have tried to bring that into your consideration. And all this happens because people are enthusiastic to work. Science is never stopping. Every day scientists are working, and practically in science, self-satisfaction is death. Personal self-satisfaction is the death of scientists. Collective self-satisfaction is the death of research. It is the restless anxiety, dissatisfaction, agony of mind that nourish science, and that motivates you to continuously do something, continuously work for people, continuously work for science with you. Okay, so I will thank all of you. I will thank the students, the eager learners with whom I am also learning. I'm learning each day and trying to learn more and more with my students. Nature, universe, only if we can combine the scientific understanding of the complex system of which we are a part, and this should happen with the awareness of the significance of our relationship to nature and universe. Then only we will learn our wholeness. So there are several things which are out of our control that redirect us to certain outcomes which are greater than those which we would have originally chosen for ourselves. So we need to clearly understand the end goal of our life, our objective, and then break it into smaller steps and then plan. This is not magic. This is how we progress. And when you try to do this, the nature and universe are with you when you are ready to do this. Thank you.
Thank you, sir, for this wonderful lecture. If anybody has any questions, they can post it in their chat boxes.
I'll stop the screen sharing.
Yes, sir.
And yes, if you have any questions, please feel free to ask.
Dr. Pradeep Sharma says it was a wonderful talk. Indeed, it was a wonderful talk, sir. You prepared a very extensive timeline from how we have reached from structure of DNA to where we are today?
In fact, I will actually appreciate Pradeep that he gave me 2 weeks, and I think I justified my effort of 2 weeks, because if this is the field bringing 70 years in
Yes.
1 hour, it's difficult. But thanks to Pradeep that at least I got 2 weeks to prepare.
No, it's thanks to you that you have presented it so well, and it is good.
It covered pretty much everything, sir. Every scientific discovery that occurred since last almost 70 years. There is a question from Santanu Sasid. He asks, can we talk? I would like to post few questions. Yes, you can definitely post questions. You can post questions in chat box. I will read for you.
Okay.
Anybody else, meanwhile, any questions?
Let him speak also, no? He can speak instead of writing.
Yeah, you can speak if you want.
Hello, am I audible?
Yeah, Shantanu, I can hear you, Shantanu. Please speak.
So that was a wonderful presentation and a beautiful representation of the timeline to the past 70 years, and you have covered almost everything that is present. I have like 3 questions towards you. The first one being on genetically modified organisms. As you had presented, the first instance being the glow pets, and the second one being the salmon fishes, and the third one being the mosquitoes that were released into the environment. But then there is a huge risk of horizontal gene transfers and vertical gene transfers across the organisms. And when we have always interfered with the nature, it has always been a backlash. It has been shown in the history. What is your take on it? That is my first question.
Exactly. I think I spoke a couple of sentences in this when I was discussing the gene drive. Kevin Eswell from MIT, when he has discovered this, and then he is saying the same thing, that without this can lead to a huge amount of ethical considerations, as well as it can have effect on the population which is naturally available, existent. So there should be rules, there should be regulation which should be drafted, and then only these things can be done. Yes, bringing them directly to the field without ethical understanding, which may be a catastrophe. So people should take time. The scientists should take time. Think about what will happen in the long term if we use it, and then accordingly they should plan the release of such organisms.
Thank you, sir. Thank you so much. My second question is on the ethical concerns regarding CRISPR technology. In one of your slides, you had put up Hu Jintao's photo. He was famous for his CRISPR babies, which he had found, which he had developed, were being resistant against HIV. There are 2 sections in the scientific society, one supporting him and one against him. Supporters say that he has done it for the betterment of the society, and there has been several instances. Even Nobel Prize winners have done Nobel Prize winners have done similar things before, like administering the drugs on themselves to fight several diseases on their sons. They have done this before, and they are comparing that incident to this. And then the other side says it is not ethically correct. What's your take on this?
Okay, so you are absolutely correct. There are people supporting him. There are people who do not support. These all are again a part of ethical guidelines are required. So in March 2019, WHO has created a committee to set the guidelines for human gene editing, and how all that committee will provide a detailed write-up, detailed module on how and how much such editing should be permitted. So I think we should wait for the WHO guideline without discussing more on that.
Okay, yeah, very diplomatic approach to that. My third question is on this. You had given the timeline based on Nobel Prize winners throughout the history. If I'm not wrong, I guess only 2 Indian names have come up. One is Dr. Corona, and the second one being Venkatrama Ramakrishnan for his ribosomal function. But then none of them are from India. They are just Indian born. What is your opinion? Like, why aren't Indians winning any Nobel? Where is the problem?
Okay. Indians are doing very good science. There is no doubt in that. We are doing very good science, and we are having government who are supporting us. The thing what I believe lacking right now in India is the high quality facilities that are being developed. They were not till few years back. It's a synchrotron came few years back only. So now such facilities should be available to Indian scientists. They should be given more freedom. Most of the Indian scientists are busy preparing the project report, preparing the utilization certificate, preparing for student presentation, statement of expenditure, purchasing chemicals, managing grants, and all those things eats up a lot of time. And more amount of flexibility should be given. Say you have been approved a project with certain objective, and now your only objective becomes to just somehow complete those objectives. You do not want to go, if you are getting some data which is something out of the line, you do not want to explore that. Fleming did that, and he developed antibiotic. If you would have an Indian scientist, he would have just kept the plate and said, no, this is not my project objective, I will not work on that. So I think giving more flexibility, leniency to scientists, facilities, government is now providing facilities, but giving more flexibility and openness is more important. And I'm sure that Indian scientists are not less than anyone else, from any other country. We will get soon.
So your answer is like the restraint which the government puts on the scientists in India is the problem.
Not only government, we should not blame government. We should not blame anyone, in fact, but it is the system. It is the ecosystem. It is expected. Say I'm in a university. I'm expected that instead of other than doing my research, I have to do 20, 30, 40 different jobs. And when research becomes one of those jobs for you, you cannot justify it. When you are completely dedicated to research, you should be given flexibility to do only research other than this the 20 other job that has been allotted. So more flexibility is required, what I believe.
Thank you so much, Dr. Timir. Wonderful talk. Thank you so much.
Thank you.
Another question is from Niharika Nag. She asks, what do you have to say about the potential use of genetic modifications for so-called designer babies?
Again, yes.
She's asking, what do you have to say about the potential use of genetic modifications for so-called designer babies?
That's the same thing, that genetically modified or where genetically modified embryo, and so that you change the babies. Again, there are huge ethical consideration. Some people will appreciate ki, okay, if you have got some identified, you can identify some genetic error, you can improve it, you can change it, and you can manipulate, you can make better better features. Say I want my son to be to play football like Ronaldo. Why should not I identify a gene of Ronaldo which is responsible for his good football skills, and then insert that gene in the fetus of so that I expect that my baby to play like Ronaldo. Again, all this comes with huge ethical considerations. It looks very good. He is very good that we can plan. We can make. I hope if I can identify the Einstein's brain gene, so that such genes everyone will want that. So again, all this comes with huge ethical considerations. Let us wait for WHO's recommendations and final comments. Then see how much they approve.
Yes, sir. I hope, Niharika, this answers your question. Any other question from anybody?
Niharika is my PhD student only. She is inquisitive, so she's...
Okay. Jennifer Rahman is saying, thank you, sir, for this informative and interesting talk. Anybody else, any question? I think there are no more questions. Thank you, sir. Thank you for this wonderful talk. Very detailed and extensive timeline you prepared, and it showed us how we reached from a simple structure of DNA to where we are, to gene therapy, to genetically modified organisms, to human genome project. Thank you so much, sir. Thank you.
Thank you again for you all, Biofootprints, and everyone present here, and the organizer for inviting me. And I'm, in fact, the seat where you are sitting, behind that, I have the first understanding of proteins I have learned in those labs only. So I am very happy to see it again, and thank you all for being